Now we are going to learn about central nervous system. So various parts of central nervous system. First we will be learning about organization of the central nervous system along with sensory system like synapses, sensory receptors, somatic sensory pathways, pain and temperature, somatosensory cortex. Then after that special senses like taste and smell along with hearing, optics of vision, the retina, visual pathways and central processing, then motor systems, introduction to the motor system of spinal cord, then cortical and brain stem control of motor function, the pyramidal tract, topics related to muscle spindle, motor reflexes, regulation of tone and posture, cerebellum, vestibular apparatus, basal ganglia, other functions and activities of brain like autonomic nervous system, functions of the hypothalamus, cerebrospinal fluid, electroencephalography and epilepsy, then sleep and the limbic system and behavior, learning and memory, then cortical function, cerebral lateralization and speech. So these topics are widely dispersed and are very elaborate. So we will be breaking down these portion into small fragments. So today we are going to learn about the organization of the central nervous system. So learning objectives include describing the basic design of the central nervous system, describing the major levels of the central nervous system function. Before learning about this topic, let's learn some major terms or jargons or technical terms related to this topic, like receptors. Receptors are specialized cells that can transduce sensory signals into nerve impulses, and subconscious activities are those activities of the body that are conducted without conscious awareness, such as the function of the heart, lungs, and intestine, or the maintenance of balance and posture. Then synapse, connection between neurons for the transmission of nerve impulse. Now we are going to learn about the topic. The nervous system is unique in the vast complexity of thought process and control of actions it can perform. Each minute, it receives literally millions of bytes of information from the different sensory nerves and sensory organs, and then integrates all this to determine response to be made by the body. Before beginning this discussion of the nervous system, then let's once again read the learning objective. Describe the basic design of central nervous system. Describe the major levels of the central nervous system function. So general design of the nervous system. Central nervous system neuron, the basic functional unit. The central nervous system contains more than 100 million neurons. A typical neuron of a type found in the brain motor cortex. Incoming signals enter this neuron through synapse located not only mostly on the neuronal dendrites, but also on the cell body. For different types of neurons, there may be only a few hundred or as many as 200,000 such synaptic connections from input fibers. Conversely, the output signal travels by way of a single axon leaving the neuron. Then this axon may have many separate branches to other parts of the nerve system or peripheral body. A special feature of most synapses is that the signal normally passes only in the forward direction from the axon of a preceding neuron to dendrites on cell membranes of subsequent neurons. This feature forces the signal to travel in required directions to perform specific nerve functions. Then sensory part of the nerve system, sensory receptors. Most activities of the nerve system are initiated by sensory experiences that excite sensory receptors, whether visual receptors in the eye, auditory receptors in the ear, tactile receptors on the surface of the body, or other kinds of receptors. Either of these sensory receptors can cause immediate reactions from the brain to memories of the experience can be stored in the brain for minutes, weeks, or years, and bodily reactions determined at some future date. Then somatic portion of the sensory system is important, which transmit sensory information from the receptors of the entire body surface and some deep structures. This information enters the central nervous system through peripheral nerves and is conducted immediately to multiple sensory areas, the spinal cord at all levels, the reticular substance of the medulla, pons and mesencephalon of the brain, the cerebellum, the thalamus, areas of cerebral cortex. Next topic, motor part of the nerve system, like effectors. The most important eventual role of the nervous system is to control the various body activities. This is achieved by controlling contraction of appropriate skeletal muscles throughout the body, then contraction of smooth muscles in the internal organs, and secretion of active chemical substances by both exocrine and endocrine glands in many parts of the body. These activities are collectively called motor functions of the nerve system, and the muscles and glands are called effectors because they are the actual anatomical structures that perform the functions detected by the nerve signals. Skeletal motor nerve axis of the nerve system for controlling skeletal muscle contraction is important. Operating parallel to this axis is another system called the autonomic nerve system for controlling smooth muscles, glands, and other internal body systems. The system is discussed later. So, skeletal muscles can be controlled from many levels of central nervous system, including the spinal cord, the reticular substance of the medulla, pons, and mesencephalon, then the base ganglia, along with the cerebellum, and the motor cortex. Each of these areas plays its own specific roles. The lower regions are concerned primarily with automatic, instantaneous muscle response to sensory stimuli, and the higher regions are concerned with deliberate, complex muscle movements controlled by the thought process of the brain. And we try to learn about the structure of a large neuron in the brain. You could see regions like dendrites, cell body will be there, then portions called axon, then synapses, then second order neurons, spinal cord will be there, and a brain connection with the dendrite portion. Next, when we try to learn about the somatosensory axis of the nerve system, we could see regions like motor cortex, thalamus, bulboreticular formation, pons, medulla, spinal cord, cerebellum, and all these related regions, and then somaesthetic areas along with some regions which are connected to this spinal cord end after pons and medulla and cerebellum region of just pons, medulla, spinal cord. That spinal cord has connections to skin, pain, cold, warmth, free nerve endings, pressure, proscenium corpuscles, expanded tip receptors, touch with the help of Meissner's corpuscles, and Golgi tendon apparatus in the muscles, muscle spindles, kinesthetic receptor, then joints. So these are the somatosensory axis of the nervous system. Then processing of the information, how that processing works. One of the most important functions of the nervous system is to process incoming information in such a way that appropriate mental and motor response will occur. More than 99% of all sensory information is discarded by the brain as irrelevant and unimportant. For instance, one is ordinarily unaware of the parts of the body that are in contact with clothing, as well as of the seat pressure when sitting. Otherwise, attention is drawn only to an occasional object in one's field of vision. And even the perpetual noise of our surroundings is usually relegated to the subconscious. However, when important sensory information excites the mind, it is immediately channeled into proper integrative and motor regions of the brain to cause desired response. This channeling and processing of information is called the integrative function of the nervous system. Thus, if a person places a hand on a hot stove, the desired instantaneous response is to lift the hand. Other associated responses follow, such as moving the entire body away from the stove and perhaps even shouting with pain. Then, role of synapses in processing information. The synapse is the junction point from one neuron to the next. Later in this chapter, we discuss the details of synaptic function. However, it is important to point out here that synapses determine the directions that the nerve signals will spread through the nervous system. Some synapses transmit signals from one neuron to the next with ease, whereas others transmit signals only with difficulty. Also, facilitatory and inhibitory signals from other areas in the nervous system can control synaptic transmission, sometimes opening the synapses for transmission and at other times closing them. In addition, some postsynaptic neurons respond with large numbers of output impulses and others respond with only a few. Thus, the synapses perform a selective action, often blocking weak signals while allowing strong signals to pass, but at other times selecting and amplifying certain weak signals and often channeling these signals in many directions rather than in any one direction. Then, storage of information, memory. We are going to learn about that. Only a small fraction of even the most important sensory information usually causes immediate motor response, but much of the information is stored for future control of motor activities and for use in the thinking process. Most storage occurs in the cerebral cortex, but even the basal regions of the brain and the spinal cord can store a small amount of information. The storage of information is the process we call memory, and this too is a function of the synapses. Each time certain types of sensory signal pass through sequence of synapses, these synapses become more capable of transmitting the same type of signal the next time, a process called facilitation. After the sensory signals have passed through the synapse, a large number of times the synapses become too facilitated that signals generated within the brain itself can also cause transmission of impulses through the same sequence of synapse even when the sensory input is not excited. This process gives the person a perception of experiencing the original sensations, although the perceptions are only memories of the sensations. The precise mechanisms by which long-term facilitation of synapses occurs in the memory process are still uncertain, but what is known about this and the details of the sensory neuron memory processes discussed later. We will be learning about that topic later. So, once memories have been stored in the nervous system, they become part of the brain processing mechanism for future thinking. That is, the thinking process of the brain compared new sensory experiences with stored memories. The memories then help to select the important new sensory information and to cancel this into appropriate memory stage areas for future use or into motor areas to cause immediate bodily response. So when we try to learn about the skeletal motor nerve axis of the nerve system, we could see regions like motor nerve to muscles, then motor area will be there, caudate nucleus region, then thalamus is there, putamen is there, I'm not sure about the pronunciation, I'm sorry, then globus pallidus, subthalamic nucleus is there, bulboreticular formation, then cerebellum, then alpha motor fiber, gamma motor fiber, muscle spindle, stretch receptor fiber. So these regions are connected with each other with the help of some connections. So skeletal motor nerve axis of the nervous system is that. Then major levels of central nervous system function. There are many levels, So the human nervous system has inherited special functional capabilities from each stage of human evolutionary development. From this heritage, three major levels of cerebral nervous system have specific functional characteristics: the spinal cord level, the lower brain or subcortical level, and the higher brain or cortical level. Then spinal cord level. We often think of the spinal cord as being only a conduct for signals from the periphery of the body to the brain, or in the opposite direction from the brain back to the body. This is far from the truth. Even after the spinal cord has been cut in the high neck region, many highly organized spinal cord functions still occur. We often think of the spinal cord as being only a conduct for signals from the periphery of the body to the brain, or in the opposite direction from the brain back to the body. This is far from the truth. Even after the spinal cord has been cut in the high neck region, many highly organized spinal cord functions still occur. For instance, neuronal circuits in the cord can cause walking movement, reflexes that withdraw portions of the body from painful objects, reflexes that stiffen the legs to support the body against gravity, reflexes that control local blood vessels, gastrointestinal movements, or urinary excretion. In fact, the upper levels of the nervous system often operate not by sending signals directly to the periphery of the body, but by sending signals to the central control centers of the cord, simply commanding the cord centers to perform their functions. Then, lower brain or subcortical level. Many, if not most, of what we call subconscious activities of the body are controlled in the lower areas of the brain, that is, in the medulla, pons, mesencephalon, hypothalamus, thalamus, cerebellum, and basal ganglia. For instance, subconscious control of arterial pressure and respiration is achieved mainly in the medulla and pons. Control of equilibrium is a combined function of the older portions of the cerebellum and the reticular substance of the medulla, pons, and mesencephalon. Then feeding reflexes such as salivation and licking of the lips in response to taste of food are controlled by areas in the medulla, pons, mesencephalon, amygdala, and hypothalamus. So in addition, many emotional patterns such as anger, excitement, sexual response, reaction to pain, and reactions to pleasure can still occur after destruction of much of the cerebral cortex, because these emotional patterns are controlled by lower brain or subcortical level of brain, like medulla and pons, etc., like mesencephalon, hypothalamus, thalamus, cerebellum, basal ganglia, all those regions. Then, higher brain or cortical level is there. After the preceding account of the many nervous system functions that occur at the cord and lower brain levels, one may ask what is left for the cerebral cortex to do. The answer to this question is complex, but it begins with the fact that cerebral cortex is an extremely large memory storehouse. The cortex never functions alone, but always in association with lower centers of the nervous system. Without the cerebral cortex, the functions of the lower brain centers are often imprecise. The vast storehouse of cortical information usually converts these functions to determinative and precise operations. Finally, the cerebral cortex is essential for most of our thought process, but it cannot function by itself. In fact, it is the lower brain center, not the cortex, that initiate wakefulness in the cerebral cortex, thus opening its bank of memories to the thinking machinery of the brain. Thus, each portion of the nervous system performs specific functions, but it is the cortex that opens a world of stored information for use by the mind. So that's all about this topic of, organization of the central nervous system. Now let's move on to the part 1, like sensory system in detail. So in this session we are going to learn about synapses. What are synapses? Learning objectives include list the type of synapses, draw a typical synapse and label its parts, explain inhibitory postsynaptic potential and excitatory postsynaptic potential, list the properties of synaptic transmission, and explain the following terms of convergence, divergence, spatial and temporal summation, occlusion, and subliminal fringe. So I'm not sure if these portions could be explained in this one video, so I will be trying. So if that is not possible, we will be continuing with another video. So let's start by learning about synapses. Let's learn some important terms associated with this topic, like Renshaw cells, inhibitory interneurons found in the gray matter of the spinal cord, that are Renshaw cells. Then second messenger, molecules that relay signals from receptors on the So information is transmitted in the central nervous system mainly in the form of nerve action potentials called nerve impulses. We know that through a succession of neurons, one after another. However, in addition, each impulse may be blocked in its transmission from one neuron to the next, may be changed from a single impulse into repetitive impulses. It may be integrated with impulses from other neurons to cause highly intricate patterns of impulses in successive neurons. All these functions can be classified as synaptic functions of neurons. So, types of synapses. Our first learning objective was to, list the types of synapses. So, types of synapses, electrical and chemical. There are two major types of synapses, like chemical and electrical. Most of the synapses used for signal transmission in the central nervous system of the human being are chemical synapses. In these synapses, the first neuron secretes at its nerve-ending synapse substance or chemical substance called neurotransmitter, often called transmitter substance. And its transmitter in turn acts on receptor proteins in the membranes of the next neuron to excite the neuron, inhibit it, or modify its sensitivity in some other way. More than 40 important neurotransmitters have been discovered this far, but we, commonly when we think of neurotransmitters, we just end up naming only one or two or three maximum. Like we will be, saying dopamine, GABA, or some other, and we will be ending like that, but that's not the actual number. More than 40 important neurotransmitters have been discovered this far. Some of the best known are acetylcholine, norepinephrine, epinephrine, histamine, gamma aminobutyric acid or GABA, in short, glycine, serotonin, and glutamate. In electrical synapses, the cytoplasm of adjacent cells are directly connected to clusters of ion channels called gap junctions that allow free movement of ions from the interior of one cell to the interior of the next cell. Such junctions were discussed later, and we won't be discussing that, but it is by way of gap junctions and other similar junctions that action potentials are transmitted from one smooth muscle fiber to the next in visceral smooth muscle, and from one cardiac muscle cell to the next in cardiac muscle. Although most synapses in the brain are chemical, electrical and chemical synapses may coexist and interact in the central nervous system. The bidirectional transmission of electrical synapses permits them to help coordinate the activities of large groups of interconnected neurons. For example, electrical synapses are useful in detecting the coincidence of simultaneous subthreshold depolarizations within a group of interconnected neurons. This enables increased neuronal sensitivity and promotes synchronous firing of a group of interconnected neurons. So, one-way conduction at chemical synapses. Chemical synapses have one exceedingly important characteristic that makes them highly desirable for transmitting nerve system signals. This characteristic is that they always transmit the signals in one direction, that is, from the neuron that secretes the neurotransmitter called the presynaptic neuron to the neuron at which the transmitter acts, called the postsynaptic neuron. This phenomenon is the principle of one-way conduction at chemical synapses, and it is quite different from conduction through electrical synapses, which is often transmit signals in either direction. A one-way conduction mechanism allows signals to be directed towards specific goals. Indeed, it's specific transmission of signals to discrete and highly focused areas, both within the nerve system and at the terminals of the peripheral nerves, that allows the nerve system to perform this myriad functions of sensation, motor control, memory, and many other functions. Then physiological anatomy of the synapse. When we just look at the structure, a typical anterior motor neuron showing presynaptic terminals on the neuronal soma and dendrites. You could see regions called dendrites, then soma region, some axons are there. That's it. So this shows a typical anterior motor neuron with dendrite, soma, and axon are the portions in the anterior horn of the spinal cord. It is composed of three major parts: the soma, which is the main body of the neuron, a single axon which extends from the soma into a peripheral nerve that leaves the spinal cord, and the dendrites, which are great numbers of horn of the spinal cord. It is composed of three major parts: the soma, it is the main body of the neuron, a single axon which extends from the soma into a peripheral nerve that leaves the spinal cord, and the dendrites, which are great numbers of branching projections of the soma that extend as much as 1 mm into the surrounding areas of the cord. As many as 10,000 to 200,000 minute synaptic knobs called presynaptic terminals lie on the surface of the dendrites. As many as 10,000 to 200,000 minute synaptic knobs called presynaptic terminals lie on the surface of the dendrites and soma of the motor neuron. About 80 to 90% of them on the dendrites and only 5 to 20% on the soma. These presynaptic terminals are the ends of nerve fibers that originate from many other neurons. Many of these presynaptic terminals are excitatory, that is, they secrete a neurotransmitter that excites the postsynaptic neuron. However, other presynaptic terminals are inhibitory, that is, they secrete a neurotransmitter that inhibits the postsynaptic neuron. Neurons in other parts of the cord and brain differ from the anterior motor neuron in 1. The size of the cell body, then in the length, size, and number of dendrites, ranging in length from almost 0 to many centimeters, then the length and size of the axon, and the number of presynaptic terminals, which may range from only a few to as many as 200,000. These differences make neurons in different parts of the nerve system react differently to incoming synaptic signals and therefore perform many different functions. Then, presynaptic terminals. Electron microscope studies of the presynaptic terminals show that they have varied anatomical forms, but most of them resemble small rounded or oval knobs and therefore are sometimes called terminal knobs, end feet, or synaptic knobs. Basic structure of a chemical synapse showing a single presynaptic terminal on the membrane surface of a postsynaptic neuron. The presynaptic terminal is separated from the postsynaptic neuronal soma by a synaptic cleft having a width usually of 200 to 300 angstrom. The terminal has two internal structures important to the excitatory or inhibitory function of the synapse: the transmitter vesicles and the mitochondria. The transmitter vesicle contains the neurotransmitter that then, when released into the synaptic cleft, either excites or inhibits the postsynaptic neuron. It excites the postsynaptic neuron if the neuronal membrane contains excitatory receptors, and it inhibits the neuron if the membrane contains inhibitory receptors. The mitochondria provide adenosine triphosphate ADP, which in turn supplies the energy for synthesizing neurotransmitter substance. When an action potential spreads over a presynaptic terminal, depolarization of its membrane causes a small number of vesicles to empty into the cleft. The released transmitter in turn causes an immediate change in permeability characteristics of the postsynaptic neuronal membrane, which leads to excitation or inhibition of the postsynaptic neuron depending on the neuronal receptor characteristics. Then, when we try to learn about the physiological anatomy of the synapse, mitochondria is here, transmitter vesicles, and in the case of presynaptic terminal, we could see mitochondria is there. Then transmitter vesicles are there, but then dendrite of neuron is on the other end, like synaptic cleft. That is the receptor proteins which are secreted into that synaptic cleft. Then next topic, mechanism by which an action potential causes transmitter release from the presynaptic terminals. Role of calcium ions. The membrane of the presynaptic terminal is called presynaptic membrane. It contains large numbers of voltage-gated calcium channels. When an action potential depolarizes the presynaptic membrane, these calcium channels open and allow large number of calcium ions to flow into the terminal. The quantity of neurotransmitter that is then released from the terminal into the synaptic cleft is directly related to the number of calcium ions that enter. The precise mechanism by which the calcium ions cause this release is not known, but it is believed to be the following. When the calcium ions enter the presynaptic terminal, they bind with special protein molecules on the inside surface of the presynaptic membrane called release sites. These binding in turn causes the release sites to open through the membrane, allowing a few transmitter vesicles to release their transmitter into the cleft after each single action potential. When the calcium ions enter the presynaptic terminal, they bind with special protein molecules on the inside surface of the presynaptic membrane called release sites. These binding in turn causes the release sites to open through the membrane, allowing a few transmitter vesicles. So when the calcium ions enter the presynaptic terminal, they bind with special protein molecules on the inside surface of the presynaptic membrane called release sites. This binding in turn causes the release sites to open through the membrane, allowing a few transmitter vesicles to release their transmitter into the cleft after each single action potential. For the vesicle that store the neurotransmitter acetylcholine, between 2000 and 10,000 molecules of acetylcholine are present in each vesicle, and there are enough vesicles in the presynaptic terminal to transmit from a few hundred to more than 10,000 action potentials. Action of the transmitter substance on the postsynaptic neuron. Function of receptor proteins. The membrane of the postsynaptic neuron contains large numbers of receptor proteins. The molecules of these receptors have two important components: a binding component that protrudes outward from the membrane into the synaptic cleft. Here it binds the neurotransmitter coming from the presynaptic terminal, and an intracellular component that passes all the way through the postsynaptic membrane to the interior of the postsynaptic neuron. Receptor activation controls the opening of ion channels in the postsynaptic cell in one of the two ways: by gating ion channels directly and allowing passage of specified types of ion through the membrane, or by activating a second messenger that is not an ion channel but instead is a molecule that protrudes into the cell cytoplasm and activates one or more substances inside of postsynaptic neuron. These second messengers increase or decrease specific cellular functions. neurotransmitter receptors that directly gate ion channels are often called ionotropic receptors, where those that act through second messenger systems are called metabotropic receptors. When we talk about next topic, that is ion channels. The ion channels in the postsynaptic neuronal membrane are usually of two types, like cation channels that most often allow sodium ions to pass when opened, but sometimes also allow potassium and or calcium ions to pass, and anion channels that mainly allow chloride ions to pass but allow minute quantities of other anions to pass as well. The cation channels that conduct sodium ions are lined with negative charges. These charges attract the positively charged sodium ions into the channel, and the channel diameter increases to a size larger than that of hydrated sodium ion. However, those same negative charges repel chloride ions and other anions and prevent their passage. For the anion channels, when the channel diameters become large enough, chloride ions pass into the channels and on through the opposite side, whereas potassium, sodium, and calcium cations are blocked, mainly because their hydrated ions are too large to pass. We will learn later that when cation channels open and allow positively charged sodium ions to enter, the positive electrical charges of the sodium ions will in turn excite this neuron. Therefore, a neurotransmitter that opens cation channels is called an excitatory transmitter. Conversely, opening anion channels allows negative electrical charges to enter, which inhibits the neuron. Therefore, neurotransmitter that opens the channel are called inhibitory transmitter. When a neurotransmitter activates ion channel, the channel usually opens within a fraction of a millisecond. When the transmitter substance is no longer present, the channel closes equally rapidly. The opening and closing of ion channels provide a means for very rapid control of postsynaptic neurons. Next topic, second messenger system in the postsynaptic neuron. Many functions of the nervous system, for instance, the processing of memory, require prolonged changes in neurons for seconds to months after the initial transmitter substance is gone. The ion channels are not suitable for causing prolonged postsynaptic neuronal changes because the channels close within milliseconds after the transmitter substance is no longer present. However, in many instances, prolonged postsynaptic neuronal excitation or inhibition is achieved by activating a second messenger chemical system inside the postsynaptic neuronal cell itself, and then it is the second messenger that causes the prolonged effect. Then there are several types of second messenger systems. One of the most common types uses a group of proteins called G proteins. The inactive G protein complex is free in the cytosol and consists of guanosine diphosphate or GDP stage plus three components: an alpha component, that is the activator portion of the G protein, and beta and gamma components that are attached to the alpha component. As long as the G protein complex is bound to GDP, it remains inactive. When the receptor is activated by a neurotransmitter following a nerve impulse, the receptor undergoes a conformational change, exposing a binding site for the G protein complex, which then binds the portion of the receptor that protrudes into the interior of the cell. This process permits the alpha subunit to release GDP and simultaneously bind guanosine triphosphate or GTP, while separating from beta and gamma portions of the complex. The separated GTP complex is then free to move within the cytoplasm of the cell and perform one or more of the multiple functions, depending on the specific characteristic of each type of neuron. The following four changes that can occur are important. Like first one, the second messenger system, by which a transmitter substance from an initial neuron can activate the second neuron by first releasing a G protein into the second neuron's cytoplasm. Four subsequent possible effects of the G protein are shown, like opening an ion channel in the membrane of the second neuron, activating an enzyme system in the neuron's membrane, activating an intracellular enzyme system and focusing gene transcription in the second neuron. Return of G protein to the inactive state occurs when a guanosine triphosphate or GTP bound to the alpha subunit is hydrolyzed to guanosine diphosphate GDP, and beta and gamma subunits are reattached to the alpha subunit. Like GDP is there, alpha, beta, gamma subunits are there. This GDP is binding into the alpha subunit. That's the structure of a G protein. That's happening when transmitter substance is usually bound to that receptor protein. After that, there will be an attachment of GTP. So that GTP will be translocating that alpha portion. So that will open channel of potassium channel usually. Then that will lead to activation of enzymes, membrane enzymes activated. Then ATP is there, CAMP or GTP, CGMP, whatever it may be, activations are happening after that. Then that activated GTP along with an alpha subunit of this G protein will activate gene transcription. That will lead to proteins and structural changes. That particular alpha portion of this G protein along with the GTP will activate one or more intracellular enzymes that will activate specific cellular chemical activators. So which are the major points happening here? Like opening specific ion channels through the postsynaptic cell membrane. We have mentioned all these points, so we'll be just reading all these points once again. So potassium channel is opened in response to G protein. This channel often stays open for a prolonged time in contrast to the rapid closure of directly activated ion channels that do not use the second messenger system. Then activation of cyclic adenosine monophosphate CAMP or cyclic guanosine monophosphate CGMP in the neuronal cell. Recall that either CAMP or CGMP can activate highly specific metabolic machinery in the neuron and therefore can initiate any one of many chemical results, including long-term changes in cell structure itself, which in turn alters long-term excitability of the neuron. Then activation of one or more intracellular enzymes. The G protein can directly activate one or more intracellular enzymes. In turn, the enzymes can cause any one of the main specific chemical functions in the cell. Then activation of gene transcription. Activation of gene transcription is one of the most important effects of activation of the second messenger system, because gene transcription can cause information of new proteins within the neuron, thereby changing its metabolic machinery or its structure. Indeed, it is well known that structural changes of appropriately activated neurons do occur, especially in long-term memory process. Then inactivation of the G protein occurs when the GTP bound to an alpha subunit is hydrolyzed to GDP. This action causes the alpha subunit to release from its target protein, thereby inactivating the second messenger systems, and then to combine again with the beta and gamma subunits, returning the alpha protein complex to its inactive state. It is clear that activation of second messenger systems within the neuron, whether they be of G protein type or another type, is extremely important for changing the long-term response characteristics of different neuronal pathways. We will return to the subject in more detail after some time, and when we discuss memory functions of the nervous system. Then next topic, excitatory or inhibitory receptors in the postsynaptic membrane. Upon activation, some postsynaptic receptors cause excitation of the postsynaptic neuron and others cause inhibition. The importance of having inhibitory as well as excitatory type of receptors is that this feature gives an additional dimension to nerve function, allowing a restraint of nerve action and excitation. The different molecular and membrane mechanisms used by the different receptors to cause excitation or inhibition include the following. When we talk about excitation, we could see some points like opening of sodium channels to allow large numbers of positive electrical charges to follow to the interior of the postsynaptic cell. This action raises the intracellular membrane potential in the positive direction up toward the threshold level for excitation. It is by far the most widely used means for causing excitation. Then depressed conduction through chloride or potassium channels or both. This action decreases the diffusion of negatively charged chloride ions to the inside of the postsynaptic neuron or decrease the diffusion of positively charged potassium ions to the outside. In either instance, the effect is to make the internal membrane potential more positive than normal, which is excitatory. Then various changes in the internal metabolism of the postsynaptic neuron to excite cell activity or in some instance to increase the number of excitatory membrane receptors or decrease the number of inhibitory membrane receptors. Now we are going to learn about inhibition. So how that is happening? Opening of chloride ion channels through the postsynaptic neuronal membrane. This action allows rapid diffusion of negatively charged chloride ions from outside the postsynaptic neuron to the inside, thereby carrying negative charges inward and increasing the negativity inside, which is inhibitory. Then increase in conductance of potassium ions out of the neuron. This action allows positive ions to diffuse to the exterior, which causes increased negativity inside the neuron. This is inhibitory. Next, activation of receptor enzymes that inhibit cellular metabolic functions that increase the number of inhibitory synaptic reaction receptors or decrease the number of excitatory receptor. Next topic, chemical substances that function as synaptic transmitters. More than 50 chemical substances have been proved or postulated to function as synaptic transmitters. Many of them are listed in the table. Let's look at them. Like neuropeptides, slowly acting transmitters or growth factors are there. So hypothalamic releasing hormones, thyrotropin releasing hormones, luteinizing hormone releasing hormone, then somatostatin, growth hormone inhibitory factor, some pituitary peptides, adenocorticotropic hormone like ACTH, then beta endorphin, alpha melanocyte stimulating hormone, then prolactin, luteinizing hormone, then thyrotropin, growth hormone, vasopressin, oxytocin, peptides that act on gut and brain, then leucine, enkephalin, methionine, enkephalin, substance P, gastrin, cholecystokinin, vasoactive intestinal polypeptide VIP, then nerve growth factor, brain drained neurotropic factor, neurotensin, insulin, glucagon from other tissues, then angiotensin 2, bradykinin, carnosine, sleep peptides, and calcitonin. These are the neuropeptides, slowly acting transmitters or growth factors. Then small molecules are there, rapidly acting transmitters, like they are of many types. Class 1 include acetylcholine. Class 2 include the amines like norepinephrine, epinephrine, dopamine, serotonin, histamine. Class 3 include amino acids like gamma aminobutyric acid or GABA, which we have mentioned earlier. Then glycine, glutamate, aspartate. Class 4 nitric oxide. So many of them are listed here, which provides two groups of synaptic transmitter. One group comprises small molecules, rapidly acting transmitters. The other is made up of a large number of neuropeptides of much larger molecular size that usually act much more slowly. The small molecule rapidly acting transmitters cause most acute response of the nerve system, such as transmission of sensory signals to the brain and of motor signals back to the muscles. The neuropeptides, in contrast, usually cause more prolonged actions, such as long-term changes in numbers of neuronal receptions, long-term opening or closure of certain ion channels, and possibly even long-term changes in numbers of synapses or sizes of synapses. Then small molecule rapidly acting transmitters. In most cases, the small molecule types of transmitters are synthesized in the cytosol of the presynaptic terminal and are absorbed by means of active transport into the mini transmitter vesicles in the terminal. Then each time an action potential reaches the presynaptic terminal, a few vesicles at a time release their transmitter into the synaptic cleft. This action usually occurs within a millisecond or less by the mechanism described earlier. The subsequent action of the small molecule transmitter on the membrane receptor of the postsynaptic neuron usually also occurs within another millisecond or less. Most often, the effect is to increase or decrease conductance through ion channels. An example is to increase sodium conductance, which causes excitation, or to increase potassium or chloride conductance, which causes inhibition. So next topic we will be learning tomorrow. Thank you for listening.